So our next two speakers is both student speakers, and the first one is Matthew Carroll, and Matthew is a graduate student working with my colleague Danny Singh in the agronomy department. So welcome, Matthew. Thank you.
All right. Thank you, Dr. Tate. Can you guys hear me okay? Yeah, okay, cool. We'll do it this way. Let's see. Oh, whoops. Maybe. All right, so my name is Matt Carroll. I'm a PhD student in Dr. Danny Singh's group, and today I'm going to talk to you about using soil maps to adjust for spatial trends within breeding trials. So there's two objectives with this project. The first one being, can we increase the accuracy of selection? This isn't unique to our method that we're proposing for spatial adjustments. Anytime you're applying spatial adjustments within your breeding program, you're hoping to have a higher correlation between your phenotypic measurement and the true genotypic value. But the second question, and this is something that when I started running spatial adjustments for our breeding program in 2020 for the Iowa State soybean breeding program, was that whenever you look at the literature, every single spatial adjustment paper talks about how they're controlling for soil fertility trends within the field, but then it's never really quantified. And there's good reason for that. Taking plot level soil data is expensive and very labor intensive. But with the advancements in high throughput phenotyping and digital soil mapping, the question is, can I map the different soil features to yield? And can we get more explainability? Can we understand what's driving these spatial trends within our field environment? Maybe. There we go. So a little bit about the breeding program here at Iowa State. Whoops, sorry about that. So as you all kind of have noticed, we'll see if it goes back. The breeding funnel, it's not new to anybody here, but here at Iowa State, we start with our A test, which would be similar to what most people would be familiar with as the progeny row stage. This is at one location. It's here just in the Ames location. This is our maximum amount of genetic diversity within our program. We then move into the B test, which would be your preliminary yield trials. This is 3 locations across the state of Iowa, but still no replication. And within these first 2 tests, we do have replicated checks, but the unique genotypes that we're evaluating do not have any replication. We then move into our C test, which would be your advanced state trials, where we have 3 to 4 locations across the state, as well as 2 to 4 replications. And then we move into the prelim and uniform trials, where this is the multi-state testing with other public universities and soybean breeders. And these are multi-state, ranging from 2 to 4 reps per location. And video didn't play. Oh well. And one thing I should say is that for our applications, we're looking at the A and B test here. So where we see spatial adjustments being the most useful within our breeding program is where we don't have replication. Once you start having replicated plots, you're able to aggregate your data, across reps. You get more accurate estimates of your yield, and so the benefits that we see this is being more in those unreplicated early generation trials for testing. So then, connecting soil maps to yield maps. So this was done in collaboration with Dr. Bradley Miller's group here at Iowa State. They do digital soil mapping. And so each one of these pixels you see is a soil feature on a per plot basis. And when I first saw this, I was really excited because this allowed us to take, samples at a 25 meter grid. They're then able to downsample this to a 3 meter by 3 meter grid, which gives us really high resolution data. And when I first saw this, I was like, yes, we're gonna be able to map soil to yield really easily. It's gonna be awesome, it's gonna be a quick project. As if you're a new grad student, if you ever say that, it's going to be a long project. It's going to take more work than you thought it was going to. And when you look at this picture here, that's the yield map of one of our early generation yield trials. And what you see is that there's maybe a little bit of correlation between the soil values and the yield, but it's not a strong correlation. There's not any single soil factor that seems to be a really strong predictor of that yield that we're seeing within our breeding trials. So then, looking at the models that we're testing, how are we evaluating this? So we looked at 5 models. The first one being our control, just if we don't do any adjustment, we just take the yield off of the combine, how are we, what are the decisions we're making? We then looked at moving means and p-splines. These are 2 methods that are traditional neighbor-based adjustments, and they're adjustments that we have used within our program, and they work fairly well. And then we looked at 2 models that incorporated the soil variability, which was the XG boost, which stands for extreme gradient boosting. It's a decision tree ML method, similar to random forest, but has a few unique differences. And the global model attempted to combine all 9 fields across 3 years and make a very generalizable model. And the local model modeled the effects of soil for each individual field. And so going back to the basics, we're all plant breeders here. We know that, phenotype is equal to your genotype plus your environment. That didn't come out very well. Sorry about that. But what we see with this then is that, your phenotype is equal to your genotype, but instead of just a general environmental effect, we're able to, quantify the micro plot that each, genotype is experiencing. So instead of environment, we're modeling the soil effect, then your population mean and some of your random error. When we first ran this, we tried to just kind of run, yield is equal to your soil, and we found that the genetic variability really confounds these results. And so one thing we were looking at with the replicated checks is that even though we have high and low yielding checks, when we started going, when we centered those check yields around the mean, they had the same range, which to me indicated that even though you have, a fixed effect of your genotype, the effect of the environment is pretty consistent across the field, meaning that it has an equal either negative or positive effect on the yields that that are being assessed. And so equation 2, we centered these data points. Now with unique genotypes, we can't center around a single genotype because there's only one. But we use the population mean, because we have a population structure in our breeding program, to get those centered variables. And the centered variable allowed us to kind of, part of that we're seeing is the true better genetic value, but part of that is going to be associated with the soil conditions that we're seeing within the field. And so then we fit that model using the XGBoost, and what we get is the new genotypic value is equivalent to the assessed phenotype, so the yield that we got off the combine, and then subtracting the soil effect. So in cases where you have a negative soil effect, your genotype is going to have an increased effect estimated, and in areas where you have a positive soil fertility effect, it's going to be estimated a little bit farther down. And what you see here in this figure is on the x-axis. That's the ground truth. That's just the initially centered variables on their population means. And on the y-axis, that's the output from the XGBoost model. And that XGBoost model, ranges. The original one is from -80 to about 40. On the predicted, effective soil, you see a range of about -40 to 20. And then lastly, how are we assessing these models, and are they doing a good job, or are they performing poorly? We looked at 3 metrics: the Moran's eye, which is a measure of spatial autocorrelation. We looked at the Cheskanowski similarity coefficient, which gives us an idea of looking at different methods, what's the true difference in the outputs. And then lastly, the mean relative efficiency, which is a measure of the ratio of the standard error, the differences between the checks. So, I like these graphs. I'm gonna play these. No. All right. And when you run your spatial adjustment, you kind of have 3 different outputs. You have an unadjusted yield, a moving mean, and XGB model. This isn't as plain as well as I'd hoped, but the unadjusted yield, when you look at this, it has a valley at the top where you see very low yield. It's non-random. When you look at the moving mean in the XGB model, you still see plots with high and low yield, but within those high and low yielding, it's not all centered in one area, so you see a more random distribution of those plot values. Which, just a gut feeling of a breeder, we expect that because we have randomized trials, we shouldn't see pockets of high and low yielding areas within the field. And so looking at the evaluation metrics, we looked at 3 different metrics. The first one being the Moran's eye statistic, that level of spatial autocorrelation. And so those first 4 box plots there are different adjustment methods. The first 2 being traditional based methods, the last 2 being the XGB model, and then the box plot on the far right being the unadjusted yield. And what you see is that all 4 methods that we tried for the spatial adjustment, they all reduced the spatial autocorrelation fairly well, but the XGB local model did have the median value across tests that was closest to 0. We then looked at the similarity coefficient. And the similarity coefficient, the best way to describe this is that if you were testing 100 lines and you were comparing 2 models and you wanted to select your top 10% of lines, if you have a similarity coefficient equal to 80%, each model would select 10 genotypes. Of those 10 genotypes that each selected, 8 of them would be identical, and each model would have selected 2 unique genotypes. That would have been the difference in what you'd seen for your selection output. And once again, what we see here is that the XGB local model does have the least similarity with no adjustment. Being different doesn't mean that it's better. It just means that there'd be less overlap between doing no adjustment and this sort of adjustment method. Then the last method we looked at was mean relative efficiency. That mean relative efficiency is a measure of the ratio of the standard error, the differences of the checks. And what we see again is that all 4 methods are better than applying no adjustment, but we see the XGB local model performs the best. And what this is telling us is that we're reducing the amount of unexplained variance when we're looking at the estimate of the genotypic effect of a line. And we're assuming that this translates not just into the checks, but also into the unreplicated lines that we're looking at across the field. So the next objective that we looked at is that how do we interpret it? So we've reduced the spatial correlation, but we still haven't talked about how do we decide what's driving the spatial variation that we see within the field. And to do this, we looked at Shapley values, which is a commonly used model interpretability method for ML. And when you look at this graph, what you're seeing is two check plots within our field, so the same genotype, but on the left, where there's a lot of blue, you see a low yielding plot, and on the right, you see a high yielding plot. And when you sum those variables that you see, it essentially sums to what the assessed value of the quality of the growing conditions were for each plot. And you can see here, this starts to allow a breeder to look at what soil interactions are driving either a high or low yielding environment within your plot, and maybe starting to try and understand and parse out, what are these interactions that we know are complex, and how are those, can we use those for further understanding of the environment that we're growing in. So I've talked about both the, how we assess the spatial side of the model. I've talked about the interpretability. Just to sum up some of our major findings, and what we're seeing is that we see an increased accuracy using the XGB local model. We see about a 77% increase in the mean relative efficiency compared to no spatial adjustment. We also see that with Shapley values, we can start looking at how do we interpret the results, what's driving the spatial variability within the field. And lastly, the way we see this being used, as we've used it in this experiment, is in unreplicated trials. That's where we see the most utility of some form of spatial adjustment, but also if you have fields that have highly variable soil conditions, this could be something that could be useful to that as well. And lastly, I'd just like to say thank you to all of our funding sources. None of this is possible without them. I'd also like to say thank you to the Same Lab staff and students for all of the work of soil sampling and harvesting that you do, as well as Dr. Miller's group for helping with soil sampling, and especially to Jonas and Shanner, who are the students who actually generated the digital soil maps for us. And if you have time for questions, I'd be happy to try and answer anything you have.
Well, let's try one question, if anyone has it. Seeing now? Well, thank you, Matt, for a great presentation.